Thank you. Thank you. So first of all, of course, I would like to thank Alex to give us the opportunity to visit this incredible botanical garden and the herbarium. And so I'm going to talk about Agrobacterium transformation and plant evolution. Now, plant evolution, you know, but maybe Agrobacterium transformation will need some introduction, and you will see in the end that the two might merge somehow, these two fields, right? Maybe we even have to invent a new word for this topic. So let me introduce myself a little bit. Where am I from and what are my interests? And so I'm actually a student of the University of Leiden, which has a very famous Hortus Botanicus that was established in 1590, okay? And you know probably the love of the Dutch people for tulips, but also many other flowering plants. Horticulture is very developed in that country. They make a lot of money, and I think it all started with the collections that came back from the colonies. You know, the Dutch, they went everywhere in the world and they collected plants and animals. They were sold, it was kind of business to sell rare things. And so they have had this wonderful collection. And you know probably that Linnaeus published his first very famous work, Systema Naturae, in Leiden. Because at that time the country was very free and you didn't have to worry about religious problems and so on. So they could publish this in Leiden. And Linnaeus came from Sweden, of course, he's not Dutch. But he later published Species Plantarum in Stockholm at that time. And that uses, for example, a lot of figures that were printed also in Leiden, as you see in the middle, right? Leiden, Latin name is Lugdunum Batavorum. That's why it's Lugbat, okay. So the Dutch were very early interested in plants, and when they had this, when they went to Indonesia, they set up a lab very early on in 1817 in Bogor, which was at that time called Buitenzorg. And they had a lab, as you can see below. This picture from 1900, they had microscopes, had everything you need. And when I was a student in Leiden, I had a professor who was called Van Steenis. And he was from Buitenzorg. The Dutch were kicked out in 1948. And so he came to Leiden. And he was very much involved in the Flora Malaysiana, which is from whole of Malaysia. And so many famous names, even in diasporas, are from the Netherlands. Probably you know. What is the Royana? Royana is from Van Rooyen, who worked together with Linnaeus. Actually, that is the origin of the name. So now I work with a botanical garden in Shanghai, which is the reason why Chen Ke is here. She was my PhD student, the last PhD student before I retired, and she now has a group there. Okay, so we try to meet every year. There's a nice team there, very young people, very enthusiastic, and they do a lot of very nice work. Okay, so we try to develop the stories that we initiated in Leiden, in Strasbourg with Ke. Systematics, as you know, is an international effort. Here is a publication from Zhang Yali, Chinese. You see there are at least 4 languages there: Chinese, Dutch, this guy from Bogor, Cohen Stewart, very important in Camellia systematics. Then in French, of course, from Vietnam, they publish in French, and in German you will also find English and so on. So that's a nice thing I feel about botany, that people can travel everywhere and meet a lot of different people. So now to Agrobacterium. So that was my first interest in Leiden. I was a PhD student there. I worked on this bacterium. At that time, we didn't know exactly how it worked. Now we have many more data. And so this bacterium, it can make tumors. Then it's called Agrobacterium tumefaciens. I don't have to explain Latin terms in a botanical department, I think. And Rhizobium rhizogenes is another one that induces hairy roots. roots that are modified. Here you see an example on tomato. It can be a problem in tomato greenhouses, right? It will make a huge amount of roots. So when people started to look at these tumors, they would make cuttings and try to see how it grows and things like that. But they also discovered very rapidly that these tumors contain specific compounds. Okay, and so here we have this mass spec that represents a kind of a symbol for the things that you can find there. And these new metabolites, these new compounds were called opines. Opine is a word that comes from octopine, from the octopus, right, where we find a similar compound, but it is a totally different thing, a totally different story. It has a different role in that organism. But anyway, these opines were rapidly discovered, and then the people found out that they can be used by Agrobacterium for its growth. So the idea is the following. Agrobacterium transfers DNA to the plant cell. This DNA is then expressed, leads to the formation of opines that are accumulating because the plant doesn't know what to do with it, exported, and the bacterium takes them up, degrades them into the original compounds. And it can survive with the help of these opines. So it's real genetic manipulation that is going on there. And to increase the number of cells that produce these opines, because only one cell typically will be transformed, there is also a system on this transfer DNA that helps to multiply these cells. That's why you have tumors. That's why you have these roots. Okay, so cell division is initiated so that the opine producing cells multiply and there will be more opines. Okay, I will say a few words about opine synthesis, because we will use it later. Here is an example of an enzyme that is encoded by these genes from Agrobacterium once they arrive in the plant cell, opine synthase. This enzyme can use different amino acids, as you see, and it will couple it with pyruvate to produce these opines, octopine, lysopene, histopine, octopinic acid, and so on. It's a process that requires NADPH. And as you can see below, 1977, I did my PhD work on the properties of lysopene dehydrogenase from crown gall tumor tissue to see how this works, purify the enzyme, study the kinetic properties, and so on. So that was how my interest for this work started, actually. Now we know already a lot about the mechanism of transfer, and I will not go into the details of that. So we have the bacterium here, the tDNA, or the region here, in yellow is the tDNA, will be transferred through a pore and then go somehow, we don't know how, into the plant cell, and then the DNA will be integrated in one of the chromosomes in a random way. Okay, that is where the DNA now will be expressed. So it's also very interesting that these genes have eukaryotic promoters, right? And they will be expressed and lead to the synthesis of the opines that we have seen before, and cell division factors that lead to the formation of tumors and hairy roots. So I already said rhizogenase induces hairy roots, and these bacteria have a very large host range. They can infect all kinds of species, right? So I want to show one example of a tDNA. That is this DNA that is transferred. Okay? There's a left border, there's a right border. And these are very short sequences, these borders, and you can take out everything that is in between and put something else. That is the basis for genetic engineering, of course. People, when they discover it, they would remove the central part and keep the borders and put their gene of interest. And now this Agrobacterium is used everywhere in the world. In each plant lab, you will find the Agrobacterium transformation as a tool. But while they were throwing away the central part, many people forgot about this central part, that it has very interesting properties. So let's have a look at these genes here. We're not going to the details, but at least the categories for these genes. There are opine synthesis genes, of course, but here you see there are 2 or 3. There are what we call plast genes in the light blue color here. What is a plast gene? Plast stands for phenotypic plasticity. it was found by a French researcher a long time ago that many of these genes are related, and what they do is they change the growth of the cells in some way. So there's a molecular mechanism that we still know very little about, and all these genes have different effects. Each gene has a different effect if you put it simply isolated in some plant. Okay. So they certainly play a role in the formation of the tumors and the hairy roots. But what exactly, we don't know. Then we have hormone synthesis genes. Here is one, and that is quite clear. It synthesizes the famous plant hormones like auxins, cytokinins. There's no problem about that. We know exactly what they are doing. And then the yellow ones, unknown function, right? These genes, we have no idea what they are doing. They're there, expressed, but what is exactly their role in tumor genesis, we have no idea. So this is just one example of a tDNA, quite a long one, from rhizogenous strain A4. Now there are many, many strains. Actually, it's an infinite variety of Agrobacterium strains. They have been there for a long time. They have been able to evolve into different structures, different organizations of genes. This is just an example. You will find other tDNAs with other tDNA genes. Okay, so that's an example. Okay, so in the beginning of these studies, 1988, is a long time ago, people thought, okay, we have these hairy roots, let's try to make plants with these, because they already knew in tissue culture how to regenerate roots into whole plants. And here we have an example of such a tobacco plant coming from a tobacco hairy root. You can immediately see, don't have to be a botanist, that this is not a tobacco plant as we know it. If you would send it to a botanical garden, they would say it's an unknown species. Okay, it's something, yeah, I've never seen before. So this is called the wrinkly HR phenotype, HR for hairy root. And interestingly, you will find it with many different species. So this touches upon something quite common in different flowering plants, apparently. So they are found in many species. The HR plants have not been studied very much after that. But maybe we're going to pick it up again. I will tell. So you can, of course, you can also study the individual genes. So on the tDNA from that we saw before, we have a gene called ROL-B. ROL means root locus, because it's kind of involved in the formation of the hairy roots. You can express it in tobacco. And so here you will see the normal tobacco leaves, and here you see the ones that have ROL-B and express ROL-B, and you see the wrinkly phenotype. So maybe the wrinkly phenotype for these hairy root plants is due to ROL-B. And you see clearly this is an inducible system, so you can switch on the gene whenever you like, and then you go from a normal plant to a wrinkly plant. So that's quite interesting, but still the question is, what is the molecular mechanism? How does the ROL-B protein do this? And I would say we still don't know after all these years. Another example from the tDNA that we have seen before from strain A4 is gene ORF13. So I did some work on that. This is the wild type tobacco. Oops, this is the wild type tobacco here. And these are different lines. Why are there different lines here? Because, like I said before, the tDNA inserts in a random way. And the expression of the tDNA genes is influenced by the surrounding DNA. So if it goes in site A, it will not be expressed the same way as in site B or site C. That's why you get a range of phenotypes for the same tDNA, right? And so we see here in this line, for example, quite nicely these heart-shaped leaves. Another example of a plast gene, phenotypic plasticity, it changes the morphology of these plants quite dramatically. Okay. Okay, now we move to the naturally genetically modified organisms, or NGMOs. That is how I will call them for the rest of the talk, right? NGMOs. So NGMOs in fact carry Agrobacterium sequences in a natural way. They are not transformed by man in a lab, but they exist naturally. And the first example that was found more or less by chance was Nicotiana glauca in 1986. in the United States. They found this, and they found the tDNA. They could sequence it already at the time. So that was the first example. At that time, people said, well, this is a quirk of nature. This is something very rare. So the idea is, of course, that the hairy roots regenerated spontaneously into a plant, a flowering and fertile plant. And that's how you got this Nicotiana glauca with its tDNA genes from Agrobacterium rhizogenes. Then, well, close to Nicotiana glauca, we have other Nicotianae. For example, Tabacum was found in '86, also carries tDNAs. And then, of course, people would say, okay, the tDNA was inserted in the ancestor of the two and inherited by these two species, so that is just not much more abnormal than what we have seen already. Now then, you see a long time between '86 and 2012. In 2012, my Russian colleague Tatyana Matvieva from St. Petersburg had the idea to look by PCR for in a big number of species from Russia and elsewhere, if there would be tDNA. And she found actually one plant, Linaria vulgaris. It has tDNA, it was sequenced, it was a very nice paper, and so they are also continuing on this type of topic. Then in 2015 came out a paper in PNAS on sweet potato. This plant is transformed. It was discovered by chance when they were looking actually at viruses in this plant, and they found some Agrobacterium sequences. And they followed it up. They didn't consider immediately it was a contaminant or something like that, but they followed it up and they could show that this Ipomoea batatas indeed contains tDNA. Okay, now you know from the sweet potato, it can regenerate spontaneously plants from the tubers. Right here you see everywhere. So you can imagine at this position here, Agrobacterium transforms a cell, and by chance, this cell will regenerate into a new plant. It will be rare, but it could happen sometimes, once in a million years. Okay. So that is still the question for this sweet potato, whether possibly opine synthesis genes from that plant lead to the sweetness of the potato. Because these opines, they are made sometimes only from sugars, so it could be that by accumulating this kind of product, you will have the sweetness of the sweet potato. But people are still looking for the opines. We know there are probably opines because there are opine genes that expressed in other plants will give you the corresponding opine. Right? But they haven't been able to demonstrate it yet in the actual Ipomoea batatas plant. So this I show only to stimulate your thinking about this problem. There are many, many steps. We will not go through them, but you see here we have, stage 1, and then other stages. So this is the first transformed. Here we see the hairy roots coming out. Here the hairy roots, they are regenerating into new plants. And in my scheme, they look different. Okay, like the hairy root plants. And then there are many steps you can imagine. And I won't go through it, but we can discuss it maybe if someone is interested in that. What could be exactly the let's say the fate of these plants and how they can survive and whether they are selected or not, right, during evolution. So in my lab in Strasbourg, we looked more in detail at natural transformation in Nicotiana. So we already knew that some of these plants had tDNA genes. You remember the glauca, it's here, this one here, and then we have tobacco, you know, probably it's a hybrid between Sylvestris and Tomatosiformis. And so we looked at Tomatosiformis, this plant, the parental, one of the parents of Tabacum, and we found no less than 4 different tDNAs. And now, these tDNAs are often inverted repeats. That means that a fragment from Agrobacterium has been inserted twice in inverted orientation, connected like that, and these sequences will start to diverge after they have been inserted. And so the level of divergence can be used to date, or to order at least, the date of arrival for these different inserts. So here we have 1, 2, 3, 4 inserts. They are conserved in Tomatosiformis. Tabacum has lost one of those, and here is another one in Otophora, Nicotiana otophora. Okay, so that this one inherits this one, the first one here, and then a second one which is specific for Otophora. You see already things are becoming more complicated, right? And so the fact that you have accumulation of tDNAs, right, kind of suggests that this process is not very rare, because how otherwise it could accumulate 4 different tDNAs. It has been transformed, retransformed, and re-transformed. It's quite amazing. So that was in 2014, and let's have a look at Tomentosiformis and Tobacum. So this is our model that we have seen before from Agrobacterium, right? With these different genes from strain A4 here. So the 4 tDNAs from Tomentosiformis are here, TA, TB, TC, TD, and you see different colors indicate that some genes are intact, other ones are not. So the black ones are the intact ones, the other ones have mutations that probably make them inactive. So you see the inverted repeats here, these arrows indicate the inverted repeats there. In all 4 we have inverted repeats. And you see maybe some things are common, like the B sequence here and the one here. And you can compare it a little bit to this one, but it's certainly not from the same strain. It's from another Agrobacterium strain. It's clearly different. And here we have something interesting. This gene called 511 was never before described in Agrobacterium. Right, so it's a new one. It must have been present in the strain that transferred this DNA. So this is a story about the repeat divergence, which permits dating of the arrival of the different inserts. And so for A, 1.2% and so on. And you can order these arrivals, these different transformation events. Okay. So let's have a closer look at tobacco. We have done some functional analysis. We are still very far from having a complete picture, of course. We're not a very big group, and we have been concentrating on the most interesting things. So we are going to look at this Tabacum, a paper of 2014. And here we have, in TB, right, we have the gene MAS2'. MAS2' is known from Agrobacterium to be a gene to produce an opine, right? Opine synthesis gene. So let's have a look at this long list, not all the details. These are tobacco cultivars, right? From, I think it's from Philip Morris originally, or French collection of tobacco plants. And the ones with the red stars, they have a very high expression of MAS2, the opine synthesis gene. The other ones, 0. So by chance, we have a nice collection of plus and minus plants, so to speak. And so you can look for the opine in a simple way. You just compare what is present in these different plants. And here we have a kind of a summary, because it's quite a long work. Here we have this MAS2 from this Agrobacterium. It will take glutamine and link it to sucrose to make deoxyfructosyl glutamine, DFG. And this DFG can also be further modified by a second enzyme, MAS1, to manapine, and with a third enzyme to agropine. And the agropine strains that make agropine in the tumors, and other ones manapine and so on. So here's some idea about the structure, right? Here we have the sugar part, here we have the amino acid part. And so we were able to show in this kind of analysis that here's the pure DFG, right? And here we have one tobacco that has high expression, and here we have one that has no expression. There is something here, but it's not at the same position. So here clearly we have the presence of DFG, and this was the first demonstration that these plants can make opines, right? Okay. Another example is Nicotiana otophora. We went to look in the herbarium yesterday, if you have it, and you have a nice collection of otophora here. It's from Bolivia, It's not a very common Nicotiana species. And so this Autophora has this specific TE fragment brought in by another Agrobacterium strain. Okay. And on this TE, oh, here's the Nicotiana Autophora, so you have an idea how it looks like, okay? And here we have quite a complex map. Actually, there are two TDs. I will not go into the details. It's too long and not very interesting anyway. But what is circled here are 3 6B genes. 6B belongs to the plast family. So you expect that if you express it in some plant, it will give you phenotype. It will change the growth of the plant. And it, we took out these 6B genes and we would study them separately. Well, we already knew from previous work that the 6B gene from Agrobacterium, if you take it from Agrobacterium, not from this NGMO plant, that it can give you very interesting phenotypes. So you put this Agrobacterium 6B gene here in tobacco, this normal tobacco, and it's some stages of the leaf development, but these are 6B tobaccos, right? And it's absolutely stunning the amount of variation that you can get. So here, this difference between this one and that one is just the insertion site of the 6B gene, right? And so this plant, I think no botanist would be able to assign it to the Nicotiana group, right? And this one also looks very strange with these tubes here. Here we have 6B flowers. This is the wild type flower from Nicotiana tabacum. These are, you see already the difference here at this position here. We have a double flower, actually, double corolla. This is called catacorollas in botany. So here we have this corolla developing in another way. Here's a very weird form. They're all based on the activity of the 6B gene. So you can imagine that when we found 6B in our Nicotiana Autophora, we got very excited because we are expecting some very strong phenotypes. By the way, can you see what happens here? Can you see? This is the same leaf here. It folds up in the lower part, and the upper part is normal, right? Something is going on here in the growth of this leaf. You can probably trace it back to the original primordia, leaf primordia and so on. But this is really fascinating. Or to find these tubes coming out here, right? It's absolutely amazing. There's another one. Okay, so we took this 6B gene from Nicotiana Autophora and we expressed it in tobacco and in Arabidopsis. Arabidopsis, the model plant, will give us much more information when we have a phenotype and so on, so it will be easier. But we also put it in tobacco. So for that we use a constitutive promoter. That means the promoter is on all the time and it's quite strong. Okay, the insertion site will be random, so we can have strong expression and weak expression depending on the lines that we get. And the transgene expression depends on the insertion site, like I said, so we can expect a whole range of phenotypes depending on where the tDNA, the 6B gene, will land in the tobacco genome. Okay, this is one example. Also here, I will not go into all the details, but this was a paper in 2018 of a tobacco leaf, which is very peculiar. What can one say about that? Yeah, it seems as if on the margins of the leaf, some new leaves are appearing, right? There's no plant like that, to my knowledge, that does this. Yeah, so it's really striking, this kind of phenotype. Then we also put it in Arabidopsis, okay? And here we have normal Arabidopsis, wild type, Thaliana, and here we have our 6B plants. And once again, we see that the leaves at the edges are strongly modified and make new leaves, okay? So that is apparently the effect of this 6B gene, and so here is an older Arabidopsis plant. It will continue growing like this, like kale, you know. It's also in the Brassica family, Brassicaceae family. And so we were able, using the phenotype and comparing it to leaf mutations in Arabidopsis, a huge collection of mutations in Arabidopsis. By comparing these things, we were able to find the mechanism, the molecular activity of this 6b protein. So the conclusion is that this protein binds TCP4 transcription factors, and we already know that TCP4 inhibits the arrest of cell division in leaf margins during development. Okay, so when the leaf starts to grow, there will be a lot of division, and then at some point division stops and the cells will just expand. It goes from here to here, just by expansion. So if you don't stop the cell division on the leaf, divisions continue and you will get these additional leaflets on the margin, right? So this was really a very important thing to find for the first time a mechanism of action of a plast protein. If you have one plast protein, you can go to the other ones, right? So from here we can extend this type of research to other plast proteins like ROL-B and ROL-C and ORF13. They all belong to this family. So that was a nice outcome of one of these genes, the study of one of these genes from the NGMOs. While we were working on this, we were also looking at other plant species, and in 2019 we published a paper on the widespread occurrence of natural genetic transformation of plants by Agrobacterium. Indeed, by looking at DNA data, we found a lot of transformed angiosperms. And so we used tDNA proteins as a query and searched NCBI whole genome sequence data. Okay, actually there are many more sequences present, but these were already assembled, so that would it make easier to find the structures of the transferred DNAs. So the estimation from this first analysis was that about 8% of all angiosperm species carry tDNA. With their tDNA genes, with their plast genes, and with the open synthesis genes. So that has quite a big, effect probably on plant evolution. Now, what do we know about this rhizogenous tDNAs from the bacteria, right? How many do we have already? Actually, not so many. If you look at this picture, I looked actually at all the NCBI data from Agrobacterium. We can only find a few types. Look here, for example, the top one, okay, with the classical tDNA type, and you see the second one, it only differs from the first one by another gene here at the end. Actually, bacterial evolution is very complex because there's a lot of lateral gene transfer, and the DNAs mix all the time. So I think after all the studies I did on bacterial phylogeny, it's impossible to make trees. Just impossible, because the mixing is going on all the time and is dominant with respect to just small nucleotide differences, right? So we can't reconstruct. We can just give the things that we find here. Okay, so there are not so many rhizogenous tDNAs known, and actually now we have more tDNAs from plants than from bacteria, because we already have a lot of tDNA structures from different plant species. So there are intact open synthase genes in the new NGMOs. I will not go exactly through this whole list, but you see some plants here, Arachis, Sidene, Dianthus, Cuscuta, Nicotiana. They have intact genes, and so you can expect opens to be present in those plants, right? These are examples for food and drink and feed plants. And so we already saw it's not on this picture. Yeah, sweet potato here. The sweet potato that made quite a big impact because people started to realize that they're eating transgenic plants, right? But there's also peanuts, guava, there's hops, kaki. That's what we are interested in now. Thanks, Alex, for your interest in our work. And we're going to collaborate on the kaki story and the Diospora story. Then we have tea, so camellia. We have wasabi, pomelo, cranberry, and so on and so on. It's a very long list. These are just a few examples. This is a nice thing. It's kind of a funny thing. Dianthus caryophyllus, you know, those beautiful flowers. It was the first flower to be genetically transformed by man to change the color, to create the blue color. So if you go to, you know, these moonlights, moon dust, if you go to a shop in France, you will not be able to find it because it's forbidden to use it. You can find it in Japan, in Australia, whatever, America. I think these new colors are available. And of course, there's a huge scope for variability created by genetic engineering. So actually, these plants were already transgenic and transformed before they were modified by man. This is an interesting story. Are there natural GMOs? So, of course, nature makes them clearly, but can one make a natural GMO in the lab? And how would you do that? So, in Denmark, a group works with wild-type rhizogenous strains and some horticultural plants. Here we have Kalanchoe, Blossfeldiana and other ones also work. And so they will wound the plant, put the bacterium there, let it do its job, get hairy roots and regenerate the plant from that. You remember the hairy root phenotype. So here we have all kinds of phenotypes. Some plants are smaller, some have more flowers, some change the color of the flower and so on. So here you see this variability that you can create by using natural strains and some natural process, right? And so is this GMO? Does this correspond to the definition of a GMO? Do you have to pay $1 million to have it tested and to feed it to rats or whatever? It's a question. It's an open question. So the Danish group says this is not GMO. We don't have to worry about it. And it seems they're already selling it in Europe. Okay? So that is kind of thing that resembles very much what happens in nature, right? It's just spontaneous, in this case, the regeneration is not spontaneous. They have to put it on tissue culture medium. Okay, so in that sense, you can say it's man-made, but of course the DNA that is inserted is not in any way manipulated or introduced in the agrobacteria, it's just there. So in case of horizontal gene transfer, everyone knows the story about the chloroplasts and the mitochondria, but now Agrobacterium seems also to play a role in this story and transfers DNA to plants, right? And it might play a role in evolution of plants. You can say this is just, you know, a chance process, doesn't do much to the plant, and we can ignore it. But it might also be that this drives some parts of the evolution of plants, right? Because it could create new forms in the beginning and then even lead to speciation, new species appearing, sympatric speciation driven by Agrobacterium. I think that is would be the most interesting thing if it is true of this story. Now we also have found some tDNA in Adiantum nelumboides, you know this maybe, a fern, pteridophyte, and the Lacaria bicolor, which is a fungus that lives in close association with plants, endomycorrhizal fungus. And this is unpublished, but this is probably very rare because we haven't come across any other examples. So this might be maybe even transfer from plants to these organisms, or for the fern, we have no idea how it happened. But it is possible that Agrobacterium has the capacity to transform other organisms than higher plants. So now let's look at camellia, because I wanted to talk a little bit about the use of these things for plant systematics. Okay, and so we have been working on tobacco, but in China they're very much interested in camellia, the tea plant, and so we decided to have a close look at the tDNAs in camellia. Family Tiaceae, subfamily 6 genera camellia, 2 subgenera Camellia, and then about 100 species according to one group, 2007, but maybe 250 or even more species according to another group. So you can see immediately there are some differences in interpretation of the data and the morphologies and the whole system. So people are clearly they are interested in establishing a correct phylogeny for these camellias. So we had started by investigating section Thea. That corresponds to about 10 species and contains Camellia sinensis. And then we investigated all of the camellias that we would get. Okay, but so let's start with the Thea section. Okay, it has 11 species. We only got 10. The other one was difficult to get. So here the names. Here we have sinensis, and in sinensis we have, of course, the varieties sinensis and sinensis assamica. Are they different? And so on. A big discussion about that, and many cultivars. So there are many sequence data on this in the databases. So what we did, I think it's put here, we have hundreds of accessions in the database, and then what we do is to recover the reads, the short fragments of 150 nucleotides that correspond to one of the to the DNA, the tDNA from section Thea. There's only one insert there, so it's relatively simple. Okay. So these reads are then used to assemble the tDNA, which is 5 kb from 150 nucleotide sequences, and we took care to separate the alleles. That is not always done when people compare sequences, right? Because it can be tricky. And we set up a new system to separate alleles together with an American company who has a software to analyze sequences, to assemble things, and now they offer also the option to separate the alleles. It can only work when the alleles are sufficiently different, okay? But it works pretty well. So we had 142 accessions, 10 of the 11 species, and we found that 60 accessions are homozygotes. They have exactly the same alleles there, and 82 are heterozygotes. Of course, for the CATA. I'm only talking about that piece of DNA. And so we have 224 alleles. With 224 alleles, you can make a tree, of course. You can construct a phylogenetic tree. And that's what we did. And here is that tree. So, okay. So what we see around here, so all around here, are different colors. They correspond to the different species from section Thea. Okay. Then we have also these dots. Can you see the blue dots? And here's some orange dots. And these dots correspond to indel sequences. So insertions from the plant genome. There are some fragments coming in from transposons or what have you that insert in the tDNA. And there are also deletions. And so these things are nicely grouped also with the sharing of these indels. Okay. Now when you look at this picture, you will note, of course, that there is a separation here. There are two groups with a separation here. So what we propose is that these two spots in fact correspond to the two CATA alleles from the ancestor of this section Thea. We now know that CATA was present well before the origin of section Thea. So that is a reasonable assumption, I think. So we have these two alleles that were present before and then generated over time all these different sequences with their different indels. Now we are going to look more in detail at B3. Okay, B3 enlarged. Here, if you look at the colors, we have 6 different species. Yes, but they're all in this group B3, and we have the same indel here, as you can see, the green one. So to me that suggests that these species share the same alleles for the tDNA, and that in fact they might not be different species at all because they share DNA. Species should be reproductively isolated and not share its alleles with another species. right? So I think I didn't want to upset too much the camellias community. I suggest it might be interesting to look again at this, using maybe other genes, other sequences and so on, so that you can figure it out, how different really these 10 species from section Thea are, or these 6 ones here. Common in there, obviously. There's a very strong evidence that they are really very similar. Now you can also look with the allele separation at what alleles combine, right? So one plant has 2 alleles, one from the father, one from the mother, and you can connect them by line. Here, that's what we did here. So you can see that A and B more or less combine here, but in part B, we see that these ones here, they always combine between B1 and B5b, and so they have this combination and nothing else, and this is Camellia quinquelocularis, and that suggests that we have a limitation in the exchange of the alleles, meaning that this is maybe a separate species, okay, that doesn't share its alleles with other ones, and only is limited to these 2 types. And you can also imagine, of course, that starting from our ancestors that we had before, we then generated, we then saw appearing this quinquelocularis at some point in time. So these have common origin, reproductive isolation probably at this level, but it's an indication. One might have to go further with complete genome sequences. We can do that now probably. How about the other camellias? We only had the ones from section Thea so far, the 10, and there are 100 or 300, we don't know exactly, but anyway there are many. So here we have the T camellias, we saw already, the flower camellias, very famous, yellow flowers, red flowers, many cultivars. The oil camellias, I don't know if you know those, but in China they can make oil with that for cooking, and also for cosmetic products. They use this Camellia oleifera. This is another one, also very nice flowers, but it's a different species and a different section even. So we were very lucky to work together with a group in Kunming. Botanical Garden in Kunming has a fantastic Camellia collection, and Professor Zhonglan Wang, he is in charge of the website for Camellia data, all kinds of data. It's a very big site, and so he is the curator of the collection. So they have the DNA well, let's say, well characterized, and we use that DNA data to extract our tDNAs. Okay, so we could map our tDNAs on their phylogenetic tree that they constructed with their DNA data. So this is in 2023. So we got these 439 Camellia accessions, 72 out of 97 species, 12 out of 14 sections, right? And we found 11 new tDNAs. So we already had the one from sinensis, CATA, and now we can add 11 more. So in this genus, transformation is relatively common, right? We see it all over the place, these tDNAs. And all of them contain one or several types of ctDNA. That was our conclusion in 2023, but we have enlarged the search by using other camellias, additional camellias, and we found some camellias that don't have tDNA. So they escaped somehow the transformation by Agrobacterium. So let's have a look at the maps of these tDNAs. This is the one from that we also find in Thea, and in section Thea, and in species sinensis. So all the T plants we looked at, they have this insert, right? So here we have TB, bigger, and all different organizations and so on. So you see the variation in sequences. It's really quite impressive, and we have also the arrows here. You see here the inverted repeats that allow us to date the arrival of these sequences. Now, the TA sequence here has been found in all the members of section Thea as heterozygote or homozygote insert. Okay. So we don't find any of the species that doesn't have a tDNA. But when we looked at the other ones, it immediately was evident that some of the members of a given species or accessions did not have an insert, whereas another accession would. That means that the tDNAs of those species, this type of tDNA, are not fixed. They have not had the opportunity yet to become generalized, and so some have it, some don't. And that means that actually when you look at one accession of a species and you don't find anything, it doesn't mean that this species doesn't have tDNA. Maybe if you study 100, you might find a few. And that means that the number of natural GMOs is underestimated, right? Okay, so here are a lot of maps, and you can see the colors once again. I think this time, we don't use the same symbols here, but if it is red in this figure, it is open reading frame, it might be active. If it's black, it's dead. Okay. And, okay, CATD within it's here, CATD, it has a very big plant sequence here, but also here the tDNA. And if you look at different sequences extracted from the databases, you will see immediately that there is not just one CATD. There are many. And this is because they have plant inserts. They are here, PL1 to PL8, different plant inserts characterize different subgroups of this CATD. So you can imagine CATD comes in, right? And then you get different lines. They will receive different plant inserts. They will also receive different, be submitted to deletion effects and so on. You can clearly see that this is not the same as over here. And so many things will go on, and they will all help you to group the species that contain this kind of modifications, right? That's also, it's like zooming in on the diversification of the plant that originally received this TDR. Now this becomes pretty complicated, but I can try to explain it. This is the Kunming camellia tree that is based on about 1,000 single copy nucleogenes. Right? They have now this tree, okay, and they have the clades here from 1 to 8, okay, this Kunming number, whatever. And here I plotted on this tree, based on the distribution of the tDNAs, the arrival, the most likely arrival time of these different forms. Okay. So here we have A to N, our 12 ctDNAs. Here we have the different types. Remember, the plant inserts, they give different types. And so if you look in detail, then you will find that this tree from Kunming is probably, it corresponds very well to the insertion patterns that we have for our tDNAs. In other words, tDNA insertion is a way to confirm in an independent way trees generated by whatever method. Right? You notice a lot of discussion whether a transcriptomics tree or a rat tree or a whole genome tree, which one is the best one, which one gives you the real result. And I always read in these papers, we need an independent way to check it. Of course, if a transcriptomics tree and a rat tree superimpose, it's okay, probably. So we propose an additional method, in fact, to construct trees. And let's see a few things. Here we have this TA coming in, because it's shared by all these guys here. And here we have the CATA number 7, the variant that we find in this group, which is the group with the 10 Thea section species. Okay, the ones we looked at before. Remember the alleles that we separated. So it's here, and it's only this part of the tree. And so it's clear from this tree that TA was already there. Okay, and you remember the proposal that we have 2 alleles to start this group of organisms here, A and B. And so here we can see it arriving in the camellia group. Okay, looks relatively simple. I mean, the picture's complicated, but you can easily find out. But the problem is that we have to look at the possibility that some of the TDNAs were deleted, because the TDNAs are modified over time, diversify in all kinds of directions. They can also be lost, and then you don't find it. So what you have to do is to look at the original insertion site to see if that is modified. So, for example, here we have TA, here we have other ones that don't have TA, and the site where it went is still intact there. So we know the intact site, and then, for example, here there might be losses of T. Let's look here. For example, here you see there is no CATA. It must have been lost. I marked it here, lost CATA. And if that is correct, this hypothesis, you should find a deletion around the original insertion site. So we have to check all of that. Right? It's a lot of work, but it's quite rewarding. It works very well. And another advantage of TDNA is that it's stable, right? And it's single copy. It will not multiply like transposer. When it's there, and it won't move around. So that was about CATA. You can see it now for the whole of the tree. And this is another one called CATM, right? It's here, you can see it here. It's only present in this group, this lower part of the tree. And these are camellias from Vietnam. And you see that actually this part, this clade, is quite different from the rest. Now, if you look in all the literature on Camellia systematics, 1958, C, monograph on Camellia, they propose that these species actually are a different genus. We call it Picretia. And so here we see Picretiana, but actually it could well be that this is a different genus. It also has received a DNA that you don't find anywhere else, and it's the only one for this group. So I think it's pretty strong evidence that this is really a separate group. So the conclusion from this work is that we have 12 insertions in camellias from 15 million years to 0.075 million years ago. This is a kind of a rough estimate on the basis of other considerations, fossils and so on. So we can kind of date that. But more important in this case is the order of arrival, right? Relative order of arrival. So the repeats of the oldest inserts have diverged by 10%. The oldest ones are CATA and CATD. They are fixed. You will find them everywhere, and they are diverged by 10% on the nucleotide. Some inserts are extremely recent. For example, this one in Camellia transarusinensis, and that's interesting because probably these TDNAs still have active genes. They haven't had the time to degenerate, to lose their activity. So you could clone the genes out one by one and see what they do on model plants, for example. Or you can CRISPR them out from the transarusinensis gene and see what happens. So many species have more than one ctDNA. We already saw that in Nicotiana, where you accumulate different tDNAs in one species. We have found transfer of inserts to man-made hybrids. So there is crossing going on between different Camellia species, and we found that the tDNAs are, of course, also transferred in the new hybrid. 10 of the 12 inserts are not fixed. So, like I said, that means that we underestimate the number of NGOs. And then we have this progressive modification of the inserts up to complete loss. And the older they are, the more likely it is that they will be lost at some point. So we have finally a very large collection of new tDNA genes in NGOs that all merit study, right? And that could be work for the next 15 years, basically, because there's so many plants, and there's so many tDNAs, and there's so many different genes, right? So here is a source for variability for not only horticultural species, but also agricultural species, wheat, whatever. Maybe we can try to use these genes as tools to influence the growth, not of the whole plant, maybe only for the flowers, only the fruits, and so on. So that is really a huge collection of the growth modifying genes. Okay, so here we have the first results on an analysis by Léon from Shanghai in the group of Ke Chen, who has looked at NCBI data for different families. And the ones in red here all contain species with TDNase. Doesn't mean that the whole family is transgenic, but it contains members that are transgenic. So it means that Agrobacterium really confirms this original finding, can transform many, many different species. It's absolutely capable of doing that, except for these monocots here. This is a group of monocots. And we know very well, of course, that monocots are hard to transform. Okay. So this is a preliminary result. And what we want to do is to make a website on the Botanical Garden from Shanghai and maybe share it with the APG4 or have a reference from the APG4 site to this specific site for NGMOs, right? And get this picture complete. But it's very hard because there are many families or orders that are huge and it takes time to go through all of that. But it's possible. So further mapping, functional analysis, we can only take a few examples probably. We can't do the whole of the thing because there are 13,000 genera, 300 species, more or less. Right? And so we have this phylogeny poster here from APG, and we have Linnaeus here once again for the systematics, original studies on plants. And so last point I want to mention is Diospores. Because of Alex, I thought I would bring the first analysis of the Diospores here to the Botanical Garden to see what we have. So it's unpublished, and we hope to add Alex also to this group of people. For sure, we already got some DNA samples and some dried leaves and things like that. So this will maybe be similar to what we did for Camellia. Because Camellia, we can still continue with the, I think we saw most of it, but now Diospores seems to be a very good candidate to continue. It has 12 different tDNA inserts so far, D, TA to BTL for 3 species. So we have Kaki, we have Lotus, and we have Olivera. They are quite close, so there are 500 or 800 species, and so there's a huge amount of possibilities there. So we looked at hexaploid plants. Now, hexaploid plants, it's very difficult to separate the alleles, right? And to separate the tDNAs. So what we did is to use the, it's in the databases, the HiFi long reads, like 50 dB, 60 dB, and they are nearly perfect. They are perfect. So you don't have to map from small reads. You have the whole thing in one go, right? So then you can separate your 6 copies per plant very easily. Right? And in that plant, that kaki plant, hexaploid kaki plant, we found 7 inserts. That's amazing. And so there are possibly 42 different inserts there in one single accession, one single plant. Some homologs show anti-sense, showing that the tDNAs are not always fixed, like we have seen before. Right? So here are the first maps for DITA. We will go up to DITM. I didn't bring it because it's not finished. It's a lot for it. And so here we have the DITA variants. It's unpublished for sure. And here we have a putative ancestor, because all these variants, right, is all DITA, they are derived from an original insert. And that has to be reconstructed by comparing all these variants. And it's not sure you will be able to reconstruct the whole original thing, because there could be a loss quite rapidly, and you will never see that piece again. So it's quite a challenge to compare these things and then to propose something like that that's most likely original insert. And then if you know that, you can draw the lines where it goes, where it comes from, what is derived from what, and so on. Okay. So this is only from Kaki. Kaki, oh, here's an Olivera one, which is close to Kaki, apparently. So this is one of the first pictures on Diospores. Okay. So that's the end of my story. I would say there's still a lot to discover. I would thank my collaborators, Thomas Fotushak, who worked on the mechanism of the 6B protein, right? Todd Blevins did a lot of bioinformatics work. Haido, who is in Kurt's lab, Wei-Ching Wang, who's a technician also in her lab, and Xiangqing Yu, who's leading the Kunming effort for Camellia systematics, the big tree I showed before. And thank you very much for your attention. Thank you.